You know that genes make up a great deal of what we are and what we will be, and that genes are in our DNA. But where exactly in our DNA? My name is Silvia, and I'm doing a PhD at the Department of Computational Biology in UNIL. In my work, I use genes to characterize the difference between species and study their evolution. For example, we know that humans share 95% of our genes with cats. To study this, we first need to know all the genes that species have. So in this video, I'm going to show you how scientists use computers to find genes. All living organisms have genes that code for protein. These proteins are responsible for how their organisms look and what they can do. For example, the protein hemoglobin, which transports oxygen through our blood and gives it its red color. All the information needed to produce proteins is in the DNA. Each DNA strand is characterized by a succession of 4 molecules, called bases, which are adenine, cytosine, guanine, and thymine, which are represented by the letters A, C, G, and T. In the cells, protein-coding DNA is first transcribed into messenger RNA, which is then translated into protein-coding sequences. We can sequence DNA to determine which bases it has and in which order they are. In recent decades, DNA sequencing technologies have improved a lot, and now it's relatively easy to know all the bases in an organism's DNA, called its genome. Once we sequence a genome, we get some very large files with millions of these 4 letters that represent the DNA sequence. It is on these very long sequences where we need to look for the genes, which occupy a very small part of them, around 1% in humans. This book contains all the letters of the smallest human chromosome, chromosome 21. ASMR. If we had a book like this for every chromosome, we would need an entire bookcase to store a genome. Imagine looking for genes through all the chromosomes of all the species that we want to study. We need a powerful computer to do this, and that's what bioinformaticians do. Program computer algorithms to perform tasks such as searching through the DNA quickly and efficiently. We know that genes start close to a specific 3-base sequence, or codon, ATG, and they finish close to 1 or more stop codons, which are TAA, TAG, and TGA. However, it's not so simple. Not all the DNA between an ATG and a stop codon is a gene. We need more information to determine if these regions are a gene or not. So what can we do? We have genes in common with other species, remember? One of the techniques used by bioinformaticians is matching genes that we know that exist in other closely related species to our newly sequenced DNA. For example, for finding the genes of a cow, you will do better matching the genes of a mouse than those of a plant, because they will have more genes in common and look more similar. It's also possible to sequence RNA. Messenger RNA is the molecule that transmits from the DNA to the cell all the necessary information to produce a protein. By sequencing messenger RNA, we get to know the sequences that genes have. Similarly to the other species genes, we can also match these sequences to our DNA to see where they are coming from. You can also tell a computer how a gene is supposed to look, like how long they tend to be or what sequences are usually found surrounding them. Then the computer will look for those kinds of regions in the DNA and return the sequences that look like a gene. Finding genes is still a difficult task that researchers are trying to improve. In the 80s, it was thought that humans had around 100,000 genes, and now we know that the number is actually much more closer to 20,000. But even today, we are unsure about the exact number. Accurately finding all the genes in a genome requires a combination of strategies. The main ones are matching genes from other species, matching RNA sequences, or using algorithms that tell us which DNA sequences look like genes. Even though it's still a very challenging task, we're getting better at it. And who knows, maybe one day you will be able to find your own genes, your dogs, or your fishes in your own laptop.